Hi everyone, I hope you're enjoying all the exciting talks at RNA 2020 in this special time. My lab is interested in this growing list of human genetic diseases that are caused by expansion of microsatellite repeat sequences in the genome. We're particularly interested in those repeat expansions that happen within non-coding parts of the genome. Today's talk is about this hexanucleotide GGGGCC or G4C2 repeat expansion within the first intron of this gene called C9orf72. This mutation causes both amyotrophic lateral sclerosis or ALS, also known as Lou Gehrig's disease, as well as frontotemporal dementia or FTD, which involves the degeneration in the cortex. The pathophysiology of C9 ALS FTD is a very active field of research. This autosomal dominant mutation has been proposed to act through either a partial loss of function or haploinsufficiency mechanism, or gain of function mechanism through the production of one or more toxic molecules, such as either the sense or antisense repeat RNA, or the polypeptides that are translated from the repeat RNA. Today's talk is about a question that we have been thinking about for some time, which is why these repeat-containing intron RNAs can persist in patient cells. As we know, after splicing, introns are rapidly debranched and degraded. Unspliced or retained introns are usually recognized as targets of cellular RNA surveillance, especially by nonsense-mediated decay or NMD, which targets mRNAs with premature stop codons. The persistence and translation of C9orf72 mRNA that retains its first intron led us to hypothesize that NMD or other RNA surveillance pathways may be compromised globally in these patients. This hypothesis makes several predictions. First, if NMD is globally defective, we would not only see an increase of the C9orf72 intron, but also in many other introns. To test this prediction, we took the existing postmortem RNA-seq data from the frontal cortex of either C9 ALS, sporadic ALS patients that do not carry the C9 expansion mutation, or control subjects. We quantify the density of reads in all the introns and compare it between patients and controls. From this distribution, we can see that the sporadic ALS patients show similar intron read densities when compared to controls. In contrast, we see a pronounced right shift or global increase of intron read densities in C9 patients compared to controls, which is consistent with our prediction. Aside from RNA quality control, NMD also regulates the abundance of a subset of perfectly spliced mRNAs. When we quantify the abundance of these previously defined regulatory targets of NMD, we again see a global increase in abundance, but only in C9 but not in sporadic ALS cases. Among these regulatory targets of NMD are the mRNAs that encode NMD factors, which is a known regulatory feedback mechanism. Consistently, we did see an increase in the mRNA abundance of several known NMD factors. Central to NMD is an RNA helicase called UPF1. But UPF1 does not only function in NMD, but also in a variety of other RNA decay mechanisms, including the degradation of this group of very special mRNAs that encode histone proteins. Unlike most other mRNAs, histone mRNAs do not have a poly-A tail in its 3' end, but instead they're stabilized by stem loop structures at their 3' end. And it is known that the degradation requires UPF1. So when we looked at the abundance of these canonical histone mRNAs, we also saw a global increase in C9 ALS, but not sporadic ALS patients. In contrast, if we looked at the histone variant mRNAs that have poly-A tails, we no longer see these increase in abundance. suggesting that the accumulation is specific to the histone transcripts that require UPF1 for degradation. Therefore, the increase in all these UPF1-dependent RNA decay targets is consistent with the idea of a global defect in RNA surveillance in C9 ALS FTD. To understand what might be the cause of NMD inhibition, we individually expressed either the sense or antisense repeat RNAs or each of the dipeptide repeat proteins or DPRs in HEC293 cells and measured the abundance of several NMD targets after 24 hours. We saw a rapid increase of NMD target abundance in cells that expressed either of the two arginine-rich dipeptide repeats, polyglycine arginine or poly-GR, or polyproline arginine or poly-PR. The effect of poly-PR on NMD targets were also confirmed when we added the synthetic PR20 peptides into the media of either mouse primary cortical neurons or human neurons differentiated from iPS cells. We saw significant increases of NMD target abundance. To further understand the mechanism by which these arginine-rich peptides inhibit NMD, we looked at the subcellular localization of UPF1 by immunocytochemistry. Normally, UPF1 is diffusely expressed in cytoplasm. In cells expressing either poly-GR or poly-PR, UPF1 is now concentrated in these cytoplasmic granules that are positive for G3BP1, which is a marker of stress granules. To test whether the recruitment to stress granules is the cause of NMD inhibition, we obtained G3BP1 and 2 double knockout cells from Paul Anderson's lab at Harvard. In these cells, stress granules induced by poly-GR and poly-PR are substantially reduced. However, the increase in NMD target abundance was almost indistinguishable from wild-type cells. We also saw very similar results when we inhibited stress granule formation using a drug called ACE-RIP. So we concluded that the concentration of UPF1 into stress granules is not the cause of NMD inhibition. Another possibility is the inhibitory effect of arginine-rich DPRs on global translation. With puromycin labeling of nascent peptides, we were able to confirm the previous observations that poly-GR and poly-PR could indeed significantly inhibit global translation. Because NMD is translation dependent, we thought that this translation repression must at least be part of the reason of NMD inhibition. To measure more quantitatively the extent to which translation repression might explain the inhibition of NMD, we treated primary neurons with a series of concentrations of either PR20 peptides or cyclohexamide, an elongation inhibitor. And then we measured the level of global translation repression and the increase of NMD target abundance. For each of the 3 targets, we saw an almost complete overlap of the dose-response curves between PR20 and cyclohexamide, suggesting that the effect of translation repression could account for most, if not all, the effect of poly-PR on NMD. Consistent with previous studies, we see that PR20 peptides are acutely toxic to cultured neurons. 48 hours after PR20 treatment, more than 80% of primary cortical neurons would undergo apoptosis and show this beating pattern throughout its cell body and neurites. Interestingly, we saw a significant increase in neuronal survival when we overexpressed wild-type UPF1, but not NMD-deficient UPF1 mutants, prior to adding PR20 peptides. This neuroprotection phenotype of UPF1 overexpression has also recently been shown by other groups in fly models expressing arginine-rich DPRs. These results suggest that the toxicity of arginine-rich DPRs is at least partly mediated by the inhibition of UPF1 and NMD. In summary, we think that a major cause of the accumulation of misprocessed RNAs in C9 ALS FTD patients is a global defect in translation and translation-dependent RNA surveillance, such as NMD. This global defect in NMD would presumably allow intron-retaining mRNAs to accumulate, including the C9orf72 mRNA that retains the repeat-containing intron 1, thereby initiating a vicious cycle that ultimately leads to neurodegeneration. We're very interested in further understanding the exact mechanism that underlies the neuroprotective effect of UPF1 overexpression. We're also curious to see whether this global deficit of translation in NMD might also play a role in other subtypes of ALS and FTD, as well as other intron repeat expansion diseases. Finally, we would really like to know why some neurons are particularly sensitive to the inhibition of these ubiquitously present RNA surveillance mechanisms. This work was led by a very talented postdoc, Sun Yu, with the help from several other lab members, including Aziz, Atta, and Jeffrey. I'd like to thank these other groups that shared reagents with us, as well as our generous funding sources. Last but not least, if you love RNA and are interested in the brain or human diseases, we have postdoc positions available. Thank you for listening.